So far in Chapter 8, we have looked at how the expression of genes is controlled by transcriptional regulators that either speed up or slow down the rate of transcription of a gene and whether or not it's transcribed at all. And as we said, the vast majority of genes in all organisms are regulated in this way. But there are a lot of other points of control. And so we're going to talk about a few of those, what we call post-transcriptional controls, which operate after transcription has begun. And we've already talked about a few of these. For example, in the figure here where it shows RNA processing after transcriptional control, we've talked about that. We've talked about how there are alternative splicings of many genes to give different gene products in different types of cells. We've also talked about the very end of the process where proteins can be controlled by additions such as phosphate groups, where they can either activate or inactivate the protein. And in the end of Chapter 8 here, we're really going to focus on this area here, talking about the control of translation. So one thing to note before we discuss this is that when we have a messenger RNA transcript and it's left the nucleus, right, it still has, as seen in this diagram down here, a 5' cap, and it has a poly A tail, and then the protein coding sequence in the middle. But a couple other things it has that will be important for this discussion is that there are 5' and 3' untranslated regions. So there's sequences around the protein, and these end up being important in control of mRNA degradation, how quickly it gets degraded in the nucleus, and also translational control. So mRNAs contain sequences that control how often or how efficiently they'll be translated into proteins. And these sequences control translation in both eukaryotes and bacteria. And the general strategy is similar for both. So these sequences can be either in the 3' or the 5' untranslated regions of the protein. This example is showing 5' untranslated region control. But in this example, showing how the RNA in that region can form a kind of a secondary structure where it's formed double-stranded RNA by complementary base pairing along that section of the RNA. And that structure that's formed is able to bind a protein called a translation repressor protein. So when that protein is in place, then no protein is made from the messenger RNA. Now note that that secondary structure of the ribosome, the double-stranded piece that's now sticking up above the messenger RNA, is right next to, there's a start codon right here on the messenger RNA, but there's also this yellow sequence which represents the ribosome binding sequence. So by controlling access of the ribosome to that sequence, the RNA can either promote or inhibit its own synthesis. Now in eukaryotes, it's a similar situation. There's the 5' cap can guide the ribosome to the start codon, the first AUG in the messenger RNA, and repressor proteins similar to those that we see up here in bacteria, this green protein. Repressor proteins exist in eukaryotes as well that can bind near the cap to inhibit translation. On the right of this figure, there's an interesting example of this type of regulation in the bacteria Listeria, and it contains a thermosensor RNA. So this thermosensor will denature at a warmer temperature when the bacterium is inside its human host. So here's the thermosensor, and when it denatures, it allows access of the polymerase to the not the polymerase, sorry, the ribosome to its binding site so that the gene can be translated and protein made. One other example of these messenger RNAs that can control their own translation in a sense is called a riboswitch. And these are similar to what we were just talking about, but just a bit more complicated as their conformation changes in the presence of a small molecule, usually a metabolite. So these RNAs have a big 5' untranslated region of secondary structure that's not too far away from what we were already talking about near the ribosome binding site. But this group of nucleotides will bind a small molecule and change the conformation of the RNA near the binding site, making it less accessible so that no protein is made. But this only happens when they're binding that small molecule, which is usually a metabolite. An example of this is in bacteria. They have a set of genes on an operon that are for purine biosynthesis. And so when guanine is one of the purines or is scarce, the riboswitch looks like this top figure here, and the guanine enzymes to create the guanine are synthesized. But when guanine is, when there's a lot of it around, I should put a plus and a minus, then guanine binds to the riboswitch. This small molecule here would be guanine and turns off the synthesis of the genes that would create or synthesize guanine. So that's a couple of examples of how translation initiation can be regulated. Another point of regulation is that the rate of mRNA degradation can be controlled, and each RNA can control its own degradation. And again, it involves the sequences in the untranslated sequences in the 3' and 5' regions of the messenger RNA. So in bacteria, most messenger RNAs only last for a few minutes before they get destroyed, and you can measure that kind of or talk about it as the half-life, the length of time that half the messenger RNA will still be around. And this allows the bacteria to adapt quickly to environmental changes if it doesn't keep its mRNAs around a long time. Eukaryotes, the half-life is longer of most messenger RNAs. Usually it's up to or less than 30 minutes, but there are some that last a lot longer. hours. But again, the shorter time they last means that the cell has more ability to change as environmental conditions change. So whether it's bacterial or eukaryotic, the mRNA's lifetime is dictated by these sequences in the 3' and 5' untranslated regions. And these regions often have binding sites for proteins that are involved in RNA degradation. Okay, the third and last type of regulation of translation that we're going to look at, let's see, that should be 3, I believe, are regulatory RNAs. So we've talked about some non-coding RNAs already and that they have various functions. For example, the transfer RNAs, the ribosomal RNAs, the small nuclear RNAs that are involved in splicing the mRNA transcript. But there's a lot of other classes of non-coding RNAs and that have recently, or semi-recently, last 15 years, been discovered. And there's a group known as regulatory RNAs that are involved in regulation of translation, and I want to talk about 3 different types of these. There's microRNAs, small interfering RNAs, and long non-coding RNAs, and they each have different roles. So we'll look at each of them in turn here. Oh, but before going on, I did want to say that I wrote this in here not very well, but that what they tend to do is form a complex with the messenger RNA through complementary binding, and they will block initiation in some situations, or they'll target the mRNA for endonuclease degradation. So basically it will get chopped up. First, we'll take a look at the microRNAs, and these regulate control of expression of thousands of genes in eukaryotes. In humans, it's thought that they regulate at least 1/3 of all of our protein coding genes. So they're a big part of regulation of gene expression. And these originate in the nucleus. So these microRNAs are transcribed in the nucleus by polymerase II, RNA polymerase II, just like our mRNAs. But they form a complex in the nucleus of this structure called the hairpin loop structure. Before they leave the nucleus, this structure is cleaved by enzymes. There's one in the nucleus called drosha, that's a big part of that. And then once it leaves the nucleus, it's cleaved again by another enzyme called dicer. Once dicer cuts the microRNA, it becomes about a 21 to 22 base pair piece of microRNA, and then it joins with an enzyme called argonaute. And that's part of this big complex of proteins called the RNA-induced silencing complex. Once the microRNA binds to that, one strand of the microRNA leaves, and the guide strand, what's called the guide strand, remains. This guide strand then searches for complementary messenger RNA and then will form a base pair complex with it. If there's an extensive match, Then the RNA gets rapidly degraded and cut into pieces. If there's not such an extensive match, then usually translation is inhibited and reduced, and later that RNA will be sequestered more quickly than others and eventually degraded. So in either case, it decreases translation or destroys and eventually destroys the messenger RNA. Now, an interesting point to all of this is that the risk is with the microRNA is released and can be used again to degrade other RNAs. So in humans, a single microRNA can inhibit transcription of a whole set of messenger RNAs as long as they have this common sequence in their 5' or 3' untranslated regions. And there's about 500 of these microRNAs encoded in the human genome. And as I said before, they regulate at least a third of all protein coding genes. Next, we'll look at the small interfering RNAs, or siRNAs. And they work very similarly to the microRNAs in terms of pairing with risk proteins, right? The RNA-induced silencing complex, and searching for complementary RNA sequences to anneal with, and then leading to the degradation of that messenger RNA. But the source of the double-stranded RNA that's used to anneal with the risk protein is different. So with the small interfering RNAs, instead of being a microRNA that's transcribed in the genome, it's a double-stranded RNA. And oftentimes these are foreign double-stranded RNAs, for example, from a virus. They can be created in our genome, nucleus in certain instances of transcription, but most are foreign at this point. That's what we understand, and they get cleaved by dicer, just like the enzyme that cleaved the microRNAs once they were in the cytoplasm. And then they combine with the risk proteins to form the complex with the risk. And once they bind, again, one strand, the guide strand, ends up staying with the risk complex and finding complementary messenger RNAs to bind with, or maybe not messenger RNAs, actually usually foreign RNAs. So this would protect the cell from invasion from, say, a virus, because it would break down that RNA. Okay, the last regulatory RNA are the long non-coding RNAs, and these are usually very long, at least more than 200 nucleotides. We think there's 8,000 or more of these in the human genome, but their role is unclear, but they do appear to be involved in silencing large sections of the genome. So just to give you an example of what we think these may do, there's one called Exist, which is involved in X chromosome inactivation. So it's 17,000 nucleotides long, huge, and it's produced by one of the X chromosomes in each cell. And remember, we talked in, I think it was, what chapter? Well, a previous chapter, maybe chapter 7, about inactivation of the X chromosome, and this is a figure from that chapter. But we talked about how one X chromosome is kind of randomly selected in each cell and inactivated, so that all of the cell's progeny from that point on has that same X chromosome inactivated. But it might be a different X chromosome in a different cell of that female, because this happens early on in development. So it's thought that exist non-coding long RNA might be produced or is produced by one of these X chromosomes, and it wraps around that chromosome and coats it, and then somehow attracts or promotes heterochromatin formation, so that piece, that X chromosome, gets very condensed and can no longer be used for transcription in the cell. The rest of the chapter, we're just going to spend a brief amount of time looking at the molecular mechanisms that create different cell types. So during development, stem cells, the kind of precursor cells in a fertilized egg, in a sense, make a decision to become one cell type or another. And this decision is is a molecular one due to the expression, right, of different types of transcription factors. So differentiation, this process of becoming one cell type versus another, is a process which the cell undergoes progressively, and it's a coordinated change to a more specialized cell type through large-scale changes in gene expression. So the figure on the slide shows a precursor or stem cell that will eventually become 8 different types of cells based on the expression of 3 different transcriptional regulators. So a wide variety of types based on a few changes in expression. The only rule in this scenario is that once a regulatory protein is turned on, it stays on through each subsequent division, so that gives different combinations of regulatory proteins, giving these 8 hypothetical cell types. And then each cell type would produce many different genes depending on that combination of transcriptional regulators. To get an idea of how important these regulators are, you might want to look at figure 816 and figure 818 in your book, because they show how using transcriptional regulators and introducing those molecules into cells can convert the cells from one form to another. So in figure 816, they're showing how they've converted fibroblast cells, or no, those were liver cells, sorry, into neuronal cells by introducing 3 nerve-specific transcription regulators. And then in figure 818, if you look at that, they're showing how they could take and introduce a set of 4 genes that each encodes a transcriptional regulator and reprogram a fibroblast cell, which is an early connective tissue stem cell, into what's called a pluripotent cell that has stem cell-like properties. So these are called induced pluripotent cells, or IPS cells, and they can proliferate indefinitely in culture. And then with specific signals, researchers can get them to differentiate into different cell types. So this is something that's being looked at in stem cell research for regenerating specific tissues or organs. So on the last cell, we talked about one of the rules of getting those 8 types of cells was that the transcriptional regulator had to be expressed and passed on to its daughter cells through each subsequent cell division. And so how does this happen? And there are mechanisms of cell memory where patterns of gene expression stay from generation to generation. And one of the most common ways this occurs is through what's called a positive feedback loop. And that's shown in this figure. And it's where a master transcriptional regulator activates the transcription of its own gene and also other cell type specific genes. So, for example, here's the gene A of the transcriptional regulator, and it makes a protein A. That's the transcriptional regulator. And when A binds to the enhancer, it creates more A protein to be created. And then when that cell divides, that protein A is distributed to all of its daughter cells. And since it's already in the cell, it will bind to the enhancer and again create more production of protein A. And so this acts as like a positive feedback loop. In each generation, that protein is passed on and then stimulates more production of itself. Okay, another method of ensuring that daughter cells remember what kind of cells they are meant to be is DNA methylation. And this occurs on certain cytosine bases in the DNA, and it's a covalent modification that generally turns off genes by attracting proteins that bind to the methylated cytosines and block gene transcription. So these DNA methylation patterns are passed on to the offspring of cells in which they occur. So here's a cytosine residue that would be one of the bases in the DNA molecule, right? And here is a methylated cytosine, so 5-methylcytosine. And again, it's a covalent long-term modification of the DNA. So this figure shows that there is an enzyme called maintenance methyltransferase that guarantees that once the pattern of methylation is present in the parent cell, that the offspring will have that same pattern. So here they're showing just an example here of a methylated cytosine on both strands of the DNA and an unmethylated cytosine. And when the DNA is replicated, getting ready for cell division, that enzyme, the maintenance methyltransferase, will interact with the DNA and recognize where the cytosines are methylated and then methylate the corresponding cytosine on the other strand so that the offspring, the DNA in the offspring, will have that same pattern of methylation. Another mechanism for inheriting patterns of gene expression involves modifications of the histone proteins. So here's a histone proteins in a nucleosomes showing that each one has a modification here in blue. And when the cell divides, the offspring is going to receive half of the parent's histone proteins with the covalent modifications. So here, that's the daughter cell receives half of the histone modifications where the other daughter cell receives the other. But there's an enzyme in cells that is responsible for recognizing, binding to the parental histone modifications and making those same modifications on histones nearby, thus repeating the pattern of histone modification that was in the parent. So all of these mechanisms that we were just talking about in terms of cell memory, they transmit patterns of gene expression from parent to daughter cell, but they don't alter the actual nucleotide sequence of the DNA. So that's why all of these are considered to be forms of what we call epigenetic inheritance. And it allows long-term changes in gene expression to be passed from parent to offspring, and kind of a permanent record of a history of what these cells have gone through.